Connaissance, recherche, sciences,
mathématiques,
théorie,
physique,
philosophie, conférence, archéologie,
nature.
Okay, well, thank you very much, Philippe, for this extremely wonderful invitation, actually, to a fascinating and important meeting. I think, really to apologize. I mean, I'm grateful for the invitation, but the organism that I'm going to talk about is a terrible example, if you like, for what we are wanting to think about here. So it's not a very important disease, Toxoplasma Gondii. It has, I suppose, the merit of being a French disease, or at least French and Brazilian disease, in that it was discovered by a great French scientist about 100 years ago. And the story that I have to tell you, which is definitely a story, tells us only that the mouse is a really terrible model for working with Toxoplasma Gondii. In fact, it's really no model at all. It's a very special situation. And perhaps the merit of what I have to tell you is to remind us all how extremely intimate, maybe we learned this from the HIV story just now, how very intimate the relations are between pathogens and their hosts. Okay, so off we go. The Toxoplasma is a malaria-related apicomplexan parasite that does its sex in cats. And the cat distributes oocysts in the feces to the environment, very persistent and robust oocysts, which are picked up by the feeding of basically any warm-blooded host is able to pick these things up, so all mammals and all birds. And warm-blooded hosts are infected by the initial sporozoites, which replicate rapidly initially in intestinal mucosal cells and thereafter in submucosal cells, in a form of the pathogen which is called the tachyzoite, or rapidly replicating form. These are not immunologically silent. The immune system recognizes something's going on. And as the immune system begins to develop a repertoire of responses, so the tachyzoites undergo a transcriptional program change to a slowly replicating form, the bradyzoite, which then goes and hides in silent tissues like muscle and brain, where they insist and replicate very slowly and persist for the life of the animal. And then they wait, essentially, until the host is eaten by the next cat. And so then the cycle continues itself. Now, one reason why, for those of us who are interested in human immunity, this is already a paradoxical situation, is that humans are very rarely eaten by cats. What this means is that that infections of humans is really end station for Toxoplasma. And that means that one can confidently say there's been no significant evolution of Toxoplasma's biology as a result of its tendency to infect humans. However, it does have an evolutionary story, and I'm going to tell you that story towards the end of my talk, but not in relation to humans. So these are Toxoplasma tachyzoites sitting on a cell and in vitro, and I want just to watch the top one as he burrows into the cell, exposing or opening up a membrane-bounded vacuole, the parasitophorous vacuole. This is in real time, so this is as long as it really takes for the tachyzoite to enter a cell. And when he's entered the cell now, if he was starting here, as it were, he adheres, he attaches to the cell, he blows something into the cell from these brown secretory organs, the rhopteries, which I'm going to talk about later on. Eventually, he penetrates the cell and forms this membrane-bounded vacuole, the parasitophorous vacuole, draws in some cellular organelles, ER and mitochondria. It begins replicating after about 10 hours, and a couple of days later, this replicating mass is released, and the new tachyzoites start another round of infection. So that's what happens. It happens to us when we're infected with Toxoplasma, and it happens to the experimental animal that I'm going to talk about, which is the mouse. In general, the fact is that the immune systems of hosts succeed in preventing extreme virulence in toxoplasma. They have, in other words, resistance. This resistance is to do with the awakening of the immune system during this early infectious stage. And I'm going to tell you now about the mouse resistance system, which is of predominant importance against toxoplasma. This is something special. You may never have heard of it before, and it probably has more to do with toxoplasma resistance in the mouse than any other infection in the mouse. So these are the molecules that do the work. They're GTPases of a distinctive family called IRG proteins. The typical RAS-like GTP binding domain, here's a triphosphate nucleotide sitting there. It's attached to this GTP binding domain, is attached to a couple of helical segments, which, among other things, can, through this N-terminal, attach to the membrane. The proteins can be myristoylated, this one is, and attached to the membrane through the myristyle group. They characteristically have about 50 kilodaltons, but there are some which consist of 2 units like this and 100 kilodaltons, and basically that's the rest of the story. They don't always use terminal myristylation to stick to membranes. There are others that use amphipathic helices. It's a family which has several members, as you'll see, and those members are non-redundantly essential for resistance of mice to toxoplasma. So normally, a mouse does not die when it's infected with a toxoplasma. This is a wild-type mouse. But if the mouse is deficient in one or other of the IRG proteins, as IgM1 here, IgM3, or the double knockout, then the mouse is as vulnerable to toxoplasma as they can be. It's as if the entire, for example, interferon system had been eliminated. So interferon gamma receptor or STAT1 deficiency will give a phenotype exactly like this. So these are non-redundantly required, even though it's a substantial family. And the necessity and sufficiency of these IRG proteins can be documented in vitro by looking here at replication of the toxoplasma during its tachyzoid phase in an in vitro system. And we're asking how efficient interferon, the main inducer of these proteins, is in preventing replication in the cells. If it's in wild-type cells, then basically the replication of these T. Gondii is completely inhibited, 100% inhibition. There's this inhibition of incorporation of a radionucleotide, a tritiated uracil. A couple of wild-type animals. For controls, if the M1 is missing, then we have a small but significant loss of inhibition. You remember in the mouse, they killed the mouse. But in the cells, we don't see such a large effect. For IgM3, it's the same. It's a bigger effect, but nevertheless highly significant. And if they're both missing, no resistance at all, the cells are completely vulnerable to the parasite. So these are essential, non-redundant resistance molecules which can function in cells. These are, in fact, fibroblasts, and in fact, most of our work is done in fibroblasts. It echoes and replicates essentially everything that we know from the mouse. So I said it's a family, and it is. In the C57 black 6 mouse strain, there are probably 21 to 23 genes encoding these proteins, and of the ones that have been knocked out, which is the red ones plus these guys, 2 of these guys down here, there is always a Toxoplasma Gondii susceptibility phenotype. They're dotted around in the genome. There's a cluster, 2 closely linked clusters on chromosome 11, another cluster on chromosome 18, and an isolated member in the top left-hand corner of chromosome 7. I will talk about these clusters again, so I'd like you to remember them. Perhaps the most interesting thing about them is that they're continuous clusters. There are no other intervening sequences in between. There's just a string of these IRG resistance proteins. So what do? They're induced massively by interferon, particularly by gamma interferon, and accumulate rapidly in cells and after interferon induction. And here are some cells that have been transfected with a GFP tagged version of IRGA6. That's the one that you saw the crystal structure of. And this toxo has just got into the cell. I'm speeding the movie up a bit, but this is what happens. The green is the GFP IRGA6. The cell is interferon treated, so it contains all of the other ones. And when we run the movie, you see the A6 basically transfers entirely from its cytoplasmic compartment, which is Predominantly, the endoplasmic reticulum is about 60% on the ER membrane, about 40% in the cytosol. So the next thing that happens is that the vacuoles disrupt. And one can see that on this move. If you look at the bottom one, toxo in the cell, the membrane just splits open at a certain point, about an hour perhaps after the loading of the IRG proteins onto the cell. And what happens next is, using the same method, but now we've got a cytosolic marker, that's just cherry, I think, in the cytosol, so a cytosolic protein marker, but also the GFP labeled IRGA6 in this cell. And what you're going to see is that, first of all, the vacuole disrupts. And then and you can see the toxoplasma as a sort of dark shadow because it displaces the cytoplasm. It's substantial and bulky, the cells are very flat. And then suddenly it disappears. It disappears because now the membrane of the toxoplasma has been disrupted. And then not just the parasitophorous vacuole membrane, but the membrane of the organism itself. And then the same thing will happen to this one. So let's watch this movie, and the first one disrupts, and then it disappears. That's because cherry goes in. Second one disrupts, cherry goes in a second, bump, and then it goes black. The movie's still running, actually. What's happening is that the cell, about an hour after the first disrupted toxo, goes into a rather unusual kind of necrotic death. So the question that it's a destructive process, and it's certainly an argument for how these IRG proteins work. But the question in our mind, and I think it's a genuinely an unclear question, is whether the primary reason, so to speak, or action of these proteins is to kill toxoplasmas, which they certainly do, or whether it's the necrotic process which releases HMGB1, which is a danger signal, a highly activating pro-inflammatory protein, which then increases the scale of the immune activity around the infecting toxoplasma. Whatever. This is what IRG proteins do. They sit in the cytosol, they assemble on the vacuole, they disrupt the vacuole, and when the vacuole is disrupted for reasons that are not clear, the toxoplasma dies, and then the cell dies, equally for reasons which are not clear. And the whole thing happens in about 2 to 3 hours. That's to say about 8 or 9 hours before replication begins in the toxoplasma. So this is the time course. We have loading, which begins within a minute or two of infection, disruption 20 minutes after the toxo dies, and about an hour after that the cell dies. Okay. So I want to talk now about virulence. We've been talking about toxoplasmas that are called avirulent. That's to say they don't kill a normal mouse. They only kill a normal mouse when the IRG system is damaged, or the interferon system which rules it. So the classical assay for virulence is how many toxos does it take to kill 100% of mice? And this is done all the time by people who isolate new strains of toxoplasma. And basically, the rule is to say that a virulent strain is one where fewer than 10 tachyzoites will kill a mouse. And an avirulent strain is one where it may be 1,000 or 10,000 or 100,000 to kill the mouse. And among the classical strains about which most work has been done from Eurasia and North America, virulence and genotype have been shown to be strongly correlated. There is a so-called type 1 strains, which are highly virulent for mice, where 1 to 10 toxos is enough, and the types 2 and 3, which are avirulent or very avirulent strains. When I say that virulence and genotype are strongly correlated, what I mean is that these strains belong to what are called clonal lineages. That's to say they have, as it were, genotypes which in package virulence, but certainly represent groups which have diverged from a population at some point in the past and then maintained a high degree of homogeneity within each group. Not all, therefore, of the differences between these strains are related to virulence. Now, it is important to say at this stage that there's been a certain amount of genetics done on virulence in Toxoplasma gondii by mating toxoplasmas in cats. And I will say right away that out of this has emerged a number of virulence factors, or virulence genes determining virulence factors, which for the time being are largely, if not exclusively, derived from the secretions from these rhoptary glands that are squirted into the cell at the moment of entry. So remember that rhoptary secretions will become important again. So what are the correlates of virulence at the cell autonomous level? We work in this is a cell autonomous immune resistance mechanism. What happens, if you like, to that story that I've just told you if the toxo that we use is a virulent strain and not an avirulent strain? And the answer is that the IRG system fails right away. That's to say, IRG proteins don't load onto the vacuoles of virulent strains. So this is a protein called B6, which shows, in this case, the most spectacular result. That's why I'm showing it to you. But here it is loading onto the vacuoles of about of two typical avirulent strains of the type 2. And you can see this loading onto more than 80% of all the vacuoles. Whereas for two virulent strains of type 1 strains, which will kill the mouse very quickly, it barely loads onto any vacuoles at all, 5 to 10%. So we see then a correlation between virulence and avirulence in the behavior of these resistance proteins of the mouse. And in a sense, when we take them away, an avirulent strain becomes virulent. Okay. So one can see that also in terms of this growth inhibition assay. I should stress that. So if we use a virulent strain, you've seen this before, then forget the multiplicity of infection, doesn't really matter. A decent dose of interferon will give complete inhibition of growth. But if we're using a virulent strain, that's to say, essentially 100% inhibition of growth, whereas there's only 40 to 50% inhibition of growth if it's a virulent strain. Of course, in terms of growth, that's an enormous difference. So indeed, virulent strains apparently resist the IRG attack, and by resisting the IRG attack, they're empowered to embark on an active proliferative episode, which essentially means, I think, that the pathogen load is higher, and when the pathogen load is higher, the immune response, I mean, going back in vivo now, the immune response is greater, and that means that the cytokine collection, which is induced by toxoplasma infection, very largely interferon gamma, but other inflammatory cytokines as well, becomes too great for the animal to handle. And insofar as it's really understood, I think one can say that an animal dying of a virulent infection or an animal which is deficient in IRG proteins dying of an avirulent infection are mostly dying of an exacerbated inflammatory response. Okay. So this really is the beginning of the end for our understanding of what's going on here, and that is that virulent strains phosphorylate one, and as we now know, at least 3 of the IRG proteins. These are immunoprecipitates of the protein that you saw the structure of, IRGA6, from a number of different strains, and what you can see is a beautiful phosphorylated band from two type 1 strains, and nothing from the group of avirulent strains that we used. So we have then a modification by phosphorylation of proteins, but only from virulent strains. What's going on here? What's happening is that 2 threonines are being phosphorylated in the switch 1 region of the GTPase that we're talking about. So those of you who are GTPase buffs, whatever you learned about RAS is essentially true of this protein as well. So this is the famous P loop here with the GGS, GKS stuff. And then this is the G3 motif, very well-defined motif. And it's these 2 threonines here which are being phosphorylated by virulent strains. In fact, what we know is that only one can be phosphorylated at a time. This is from mass spectrometry. So either this one or this one is phosphorylated. There seems to be a reason for that. And that is that these 2 threonines are in the switch 1 loop, which is modified, remodeled by the binding of nucleotide. And it appears in the various crystal structures that we have for A6, it's really remarkably flexible. Either the loop has this little helical turn in it, which turns T108 out towards the environment, and T102 looks down, that's a magnesium ion. Or in another structure, we have T108 pointing down towards the gamma phosphate, and the T102 is looking in the other direction. It seems as if somehow these 2 threonines are alternatively in that position or in that position, and if they're in that position, one of them gets phosphorylation, in the other position, the other one does. Whatever, that seems a reasonable position right now. What about these threonine phosphorylations? Well, what we did was, of course, to make phosphomimetic mutations of these threonines by replacing the threonine with aspartic acid. And in this assay, we are looking at the ability of these proteins to hydrolyze GTP, which is an essential part of their function, I should say. I didn't go into all that. And what we can see is that both T102D and T108D completely block the hydrolytic function of the GTPases. And we also made the neutral alanine mutations that have the same effect. So the wild-type protein hydrolyzes GTP, more or less well. It's a lousy GTPases, but nevertheless that's essential for their function. And these phosphorylated threonines, or at least the phosphomimetic versions of the threonines, destroy that activity. Okay. So, I mentioned to you that virulence genes had been located to secreted proteins from the ruptures of virulent strains. And it was obvious to us, and it's for those of you who know the field, it's obvious to you, that there was something very important about the fact that several of these things are kinases. So once we had all this kind of data, we asked David Sibley, who is both colleague and competitor in this business, I should say, to give us, with his name on them, 3 strains of an avirulent strain, 3 lines of an avirulent strain, a type 3 strain called CTG, which are transgenic either for an active kinase from the ruptures or for an kinase dead version of the same protein, a mutant, or for the vector alone. So these 3 strains, which you see here, that's the vector alone, that's the kinase dead strain, and that's the active strain. And we infected interferon-treated fibroblasts with these 3 strains and immunoprecipitated A6. And what we find is, indeed, that the active kinase can phosphorylate A6. What this means is that a CTG, an avirulent strain, secreting this ROP18 kinase, can phosphorylate IRGA6 in infected cells, and we know that that will inactivate the GTPase. Furthermore, it deposits, or allows to deposit, phosphorylated IRG6 on the vacuole. One can see that here. Always look at the red dot runs. That's what's really interesting here. This is the active kinase. That about 90% of the vacuoles on this strain are covered in phosphorylated IRG6. We know it's phosphorylated because we have made antisera against the phosphopeptides. So this is against phospho-102 and phospho-108. Whereas essentially nothing's happening for the vector alone or the phosphate dead version. It's also true that the amount of phosphorylated stuff that we can detect is greatly increased when infections with the active kinase CTGs. And lastly, and most importantly, in a way, the total amount of non-phosphorylated IRGA6 drops when the animal is infected with the active, with a avirulent strain secreting the active kinase. So that there's phosphorylated material, which you know is inactive, but the total amount of active material that's on the vacuole drops a substantial amount, almost to background, from the levels in from vacuoles from strains which are not carrying the active ROP18 kinase. So just to say, how do we know this? Well, we have an antibody, monoclonal antibody, which recognizes only the GTP-bound form of the IRGA6. And so this is strikingly reduced as the phosphorylated IRGA6 goes up. And we know that the phosphorylated IRGA6 is unable to enter the active form. And so that's somehow there's a reciprocation between these 2 results. But what's interesting, you can see, is also that this is only true, if you may be able to see it, very low interferon concentration. So only if we use really a very small amount, 0.3 units, do we see this effect. There's a modestly significant effect at 1 unit, and at 3 units, it basically disappeared. Now, the difference between virulent and avirulent strains is something we can see at 100 units of interferon. So there's more. It could be a pure expression thing. This is a synthetic system, but it could also be, and as we believe, there are multiple virulence proteins involved, and we're looking at only one of them. Okay. Just looking at pretty pictures, this is IgA6, and then this is the phospho-102 staining these 2 vacuoles very nicely. You see this one here can't be seen by the antibody that sees the active form, even though it's easily seen with PT102. And so it goes on. I won't go through that in the interests of time. Okay, now, so we have a little story then that the mouse can make these proteins which make the difference between life and death for avirulent strains. Virulent strains can defeat them. They apparently defeat them by phosphorylating the IgG proteins and eliminating their function. But virulent parasites are a funny thing. If they kill mice in 10 days, then that seems like a bad policy. It's bad for the mouse because it drops dead in 10 days, and it's bad for Toxoplasma because it essentially fails to complete its infectious cycle. The mouse gets sick, drops dead, end of story, and the next cat is too late. And it should be highly disadvantageous in what we call the cat-mouse turbo cycle. I'll come to that in a moment. So we've asked the question, maybe the mouse IRG mechanism is itself polymorphic. We have polymorphism in the Toxoplasmas. Some are virulent, some are not. ROP18, I should say, differs substantially in sequence between the three types of Toxoplasma. And maybe type 1 strains are only virulent for some mouse genotypes. So that turns out, in fact, to be the case. But let me just say something which is profoundly important for understanding the toxo situation, and that is that Toxoplasma has been involved in a very dramatic evolutionary transition in the last 10 to 15,000 years. This is how it started. A felid as the sexual host, any warm-blooded animal as the intermediate host. And in the old days, it was moving between any true felid, any real cat, by using the fact that cats tend to eat rather indiscriminately warm-blooded prey, so that a toxo that was infecting a lynx could move into a leopard or vice versa from to a wild cat that was basically free to move because of the overlap in the prey species. But if you are a parasite that requires horizontal transmission, then cats are the worst imaginable hosts because they're the top of the food chain. And what that means is that within any mammalian community in the wild, the high-end carnivores are always the rarest species around. So not only are they rare, they also are normally solitary species. They hunt alone. They define a large territory in which they hunt, and they try not to meet if they possibly can. Bad for transmission. So it's clear that the prey species are doing the transmission for them. It's not a bad technique, as it turns out, because Toxoplasma, unlike many of the organisms that we've been talking about here, is one species worldwide. A single species. So it has succeeded using this interesting strategy in exploiting the fact that the cat world is basically worldwide, but jumping from species to species in a kind of leisurely way. Many things about Toxoplasma biology suggest that it's not in a hurry. It just sits in the brain and waits, or sits in the environment as an oocyst and waits. That's how it was. But 10,000 years ago, we invented agriculture. And when we invented agriculture, we invented food surpluses. And when we created food surpluses, we created an opportunity for a certain class of commensal animal, the mouse and the rat, these small rodents whose population numbers began to increase vastly inside human communities. And cats in Asia Minor were domesticated from a single subspecies of Felis sylvestris and became the domestic cat. And the domestic cat has essentially replaced all wild cats as the evolutionarily significant sexual host species for Toxoplasma Gondii. Essentially, all the rest of them are basically irrelevant, and any evolution which is going on in Toxoplasma Gondii is going on in this relationship here between the cat and mouse, of course, one or two other small rodents and small birds that are caught, but the mouse is also a global species now and therefore of considerable interest. And indeed, I want to draw your attention to the fact that both the cat and the mouse only reached Brazil about 500 years ago. So there's some very interesting new science to be done there. Okay. So we were looking for polymorphism, and we took a strain of mouse called Casti IJ, which comes from Thailand. And what we discovered by sequencing both of the main IRG clusters is that the black 6 genome is this very long one here, the Casti IJ is this one here, and 1, 2, 3, 4, 5, possibly even 6 genes are simply missing. So it's an incredibly dynamic chunk of chromatin. And the next one is chromosome 18, is the same story. There's in fact only one, what we think is a functioning IRG gene in the Casti IJ chromosome. And here a whole lot are simply not there. Casti IJ is reasonably competent to deal with Toxoplasma Gondii. So very dynamic, already telling us that we're dealing with a polymorphic system. And so we've sequenced a lot of IRG proteins from wild mice from around the world. And what we see is that there are some which are really very conservative, like M1, only a single amino acid variant over this group of mice. Out of 408 amino acids. And so it goes until we get around here to IRGB6, which is incredibly polymorphic in the wild. So more substitutions of amino acids here than between a typical pair of classical class 1 MHC molecules. 46 out of 415 residues are variable in the wild population. And we know from the domestic mouse that this is an active resistance protein. So we became interested in this question and wondered whether this polymorphism might have some kind of biological, or in polymorphism in general, might have some biological relevance. And we focused our attention on a mouse called Sytem, which is maintained by François Bonhomme in Montpellier, but actually comes from southern India. And we discovered to our delight that of three strains that we tested, three wild strains that we tested, Sym is completely resistant to virulent strains of Toxoplasma Gondii. So I show this using, again, this inhibition of uracil incorporation. These are cells taken from the diaphragm. That's how we collect cells from other strains. So if we take the classical inbred strain, just to remind you, then basically it's 95% inhibited by interferon treatment in the classical inbred strain for the avirulent toxo, and only about 30% for the virulent toxo, that's what I said before. But with the Sytem strain, it's 95% blocked for both the virulent and for the avirulent toxoplasma. In other words, we do indeed have a mouse polymorphism which appears to, if you like, respond to the virulence polymorphism in the Toxoplasma Gondii. And although we haven't worked this out yet, I just want you to look at this picture, which is a virulent strain. It's RH with a YFP tag in it, and another IRG protein, which appears to be very important for some reason or other for this strain of mouse, beautifully coating 6 out of 6, and in fact it's over 90%, over 95% of vacuoles are coated by this IRG protein, something we've never seen, of course, in the laboratory mouse. So there's a world of discovery for us in terms of working it out. But I think that the issue that I want to bring to your attention is that these diseases exist in a world of complex outbreeding hosts, and that the question of resistance and virulence and so forth is not really something that one can discuss without discussing the genetics of both participants. Okay, so our conclusions. The IRG proteins are an essential resistance system for mice, but Toxoplasma Gondii is polymorphic, and type 1 strains are highly virulent because they express the kinases that can inactivate IRG proteins. But the IRG system is highly polymorphic in wild mice, and there are some IRG alleles that can resist virulent T. gondii. And this looks like a pretty dynamic situation. You have to realize that this is a situation which is only about 10,000 years old. This is a new evolutionary situation that the mouse has got, mouse and Toxoplasma and cat have got themselves into. Now, I haven't talked about vaccination, but this is, I think, a veterinary problem here, and it's one that, frankly, has not been given the attention. That's unfair to those people who have tried to look at this, but it has not really been given the attention it deserves. If we want to get rid of Toxoplasma from the Western world, then what we have to do is to concentrate on learning how to vaccinate cats. Because we can interrupt the domestic cat is essentially the only important vector now for the reproductive vector of Toxoplasma Gondii. And although there have been various attempts, there's no real enthusiasm for this project at present, and I would say if we want to deal with this, then this is the way where we have to start. Okay. So many colleagues in my own lab, some still there, some no longer still there, mass spectrometer colleague locally, several colleagues in the outside world. Gabi Reichmann was the person who first introduced us to Toxoplasma not that long ago, and the crystal structure of IgA6 we did with Eva Wolf in the Max Planck Institute in Dortmund. And thank you all very much.